Hi, welcome to another session in the Healthcare of the Client 3 course. In this session, we are going to continue our discussion by focusing on the concept of genetics. Before we proceed, please take time to review the objectives for this concept. Genetics is essential in medical practice and nursing because it provides the tools to determine the hereditary component of many illnesses and disease. It also improves our ability to predict their susceptibility, onset, progression, and response to treatment. However, there is a gradual shift from genetics to genomics. So what are the differences between genetics and genomics? Well, let us look at their definitions. While genetics is the study of the general mechanism of heredity and variation of inherited traits or characteristics, genomics focus on the structure and function and evolution of the DNA and how the mapping of genomes or these genes affect disease development, prognosis, and treatment. It doesn't matter your age, socioeconomic status, race, or ethnic background, every individual can be affected by a genetic disorder. Nurses play an important role in genetics and genomics. This is because there are actually nursing specialty areas in the field of genetics, and nurses are very much involved in the education and counseling of clients. As advancements continue, nurses are assuming an expanded role in areas such as preconception counseling and testing. That is where you give counseling to couples who are ready to start reproduction or to start having babies, to determine their genetic risk and how to be able to avert any genetic disorders in their infants. There is also neonatal genetic screening and testing, and this is after the fact when a baby is born that nurses are able to put together the genetic history, predispositions, and risk factors to educate the client on how to take care of this baby. Palliative care for infants with life-threatening conditions is another aspect of the expanded role of nurses. Palliative care usually encompasses comfort care, and once you know the genetic predisposition, you can begin planning for that ahead of time, even before the baby is born. Another role that nurses assume is the identification and care of individuals with genetic conditions such as cystic fibrosis or any other conditions that may be there. Specialized care of women with genetic conditions during pregnancy is also another role, and this includes congenital heart disease. We will talk about the tetralogy of Fallot, cystic fibrosis, and factor V-laden illnesses, and these are just some of the examples. To be able to provide care in the field of genetics, a nurse must possess some essential competencies, and these competencies have been developed by various bodies of nursing organizations. And basically, nurses must have working knowledge of genetics and genomics to be able to construct a pedigree from collected family history information. So in the hospital, it is recommended that with every admission you get the genetic history of that particular patient or client, so you can piece together to know exactly what the risk for this individual is, so you can begin educating the client. Nurses also have to be able to develop a plan of care that incorporates genetic assessment, and this is part of getting the genetic history and planning to educate the client on their risks. Then they should be able to also provide patients with genetic information, resources, and services. So what resources in the community? Are there any groups that will be supportive to the patient? Are there any federal or state resources for those conditions such as cystic fibrosis or for diabetes or anything that the client may be predisposed to? Then nurses have to be competent in genetics to be able to facilitate referrals for specialized services and also be able to evaluate the effectiveness and impact of genetic and genomic technology, intervention and treatment. So there are various treatments that are out there, and nurses should be able to have a working knowledge, if this is the field you choose to be in, to be able to refer your clients to the appropriate facilities for testing and also for counseling and treatment of certain genetic conditions. The competencies developed will help nurses to function in the performance of the roles that we have talked about, including the prenatal screening and counseling, career testing, such as if a patient or a client is a carrier for a certain genetic disease such as sickle cell, newborn screening to identify genetic conditions for treatment, palliative care, and all the other roles that we talked about in the expanded role for nurses. So according to the NIH, the Human Genome Project was an international scientific research project to sequence and map all the genes of humans. And together this is known as the genome, human DNA. The project was completed in April 2003 and has resulted in many advances in genetic testing. Some examples of the benefits from this project include genetic testing. There are now direct or molecular testing that can be done. There is linkage analysis that can link certain genes to certain disease, and biochemical testing. There are also prenatal tests that has been developed as a result of this, and this include maternal serum screening, which can pretty much identify certain genetic disease in the early term of the pregnancy. There is also the fetal ultrasound, amniocentesis, chorionic villus sampling that we will be talking about when we talk about pregnancy. So all these tests will help the nurses and providers to be able to come up with a plan of care which will benefit the client by limiting the effect of these genetic conditions. And so when it comes to genetics, there are various ethical, legal, and social implications because there is the concern for privacy and fairness in the use and interpretation of this genetic information. For example, there are cases where the law enforcement bodies are now using genetic tests to actually identify people who committed crime. So where does the boundary stop? If I go in for genetic counseling or genetic testing, can law enforcement actually retrieve my genetic information by issuing a subpoena to that facility I went to for them to release my information to them? And so that is a privacy concern. Also, there are social implications. What happens if everybody is testing for their genes, and are there going to be gene modifications and all that? There is also the problem with clinical integration of new genetic technology. So genetics are fairly new, and we are just beginning to understand the extent to which we can use genetics. So what happens if there is not a clear integration of these genetic technologies that we are using? What about possible misuse, discrimination, and stigmatization when We know for a long time our society has been plunged with discrimination and all kinds of things. What if genetics allows us to find a particular gene that is believed to be dominant and that is resistant to disease and all that, and only a few people can benefit from it? So all these are legal and ethical conditions that can affect genetics. Factors influencing decision to undergo genetic testing are seldom autonomous. That means that family members can actually push other family members to go for testing. For example, if I know that I might be at risk for a certain genetic condition, I might ask my parents to go for testing so we will know exactly what we are at risk for, although they might not want to do it. Okay, they are also based on feelings and of commitment to others. Like my parents may be feeling bad because they think they are predisposing me to a genetic condition that might coerce them to go for testing, and that is as a result of their commitment to me or to my other siblings. There are also socioeconomic factors. Many people cannot afford to do genetic testing because it's still very much expensive, and as such, there will be inadequate sharing of the resources that we have with regard to genetics. There are also cultural and ethnic differences. Some people culturally are scared to go for some of these tests, others are liberal and want to go for these tests. And genetics can also strain family relationship. Example, if I know that my parent tested, or if you find out that family member tested and have this condition, you may be asking, why should you go for this testing and place this burden on us that we are now going to be carrying this burden of knowing that we can potentially have this disease? So this can strain all forms of relationship. In the marital relationship, genetics can also be a factor of strain, because if I find out that, say, I have sickle cell and my wife also has sickle cell, then that means that we may decide, or I might decide, I don't want kids because I don't want my children to have sickle cell. But my wife, on the other hand, may think that she wants kids, or maybe I may say I want kids. My wife will say I don't want any kids because I don't want them to have sickle cell. And that can cause a strain in our relationship. Genes are the coded instructions for the cellular regulation of all the proteins the human body produces. So when we talk about clinical genetics, we are talking about the genes, chromosomes, DNA, and everything that makes up the genetic of an individual. So these genes regulate all the production of proteins in the body, and genes are divided into specific large chunk or highly condensed double-stranded DNA known as chromosomes. Humans have 46 chromosomes divided into 23 pairs, and the DNA replicates and then organizes these chromosomes into a chromosome form when the cells are dividing to ensure precise delivery of the genetic information to each of the two new daughter cells. And if there are any abnormalities happening with this, then there is going to be a genetic disorder. So the phenotypes of any gene for a person is the characteristics that can actually be observed, or in some cases determined by a lab test. And then the genotype for a person's single gene trait is what the actual alleles, what we call alleles, are for that trait, not just what can be observed. So the gene or the genotype is what the actual trait is, or is what the alleles that are portraying the actual trait is. There are two types of alleles. We have dominant and then recessive alleles. And these determine gene expression. So for every single gene trait, a person inherits one of these alleles for that gene from the mother and one allele also from the father. A dominant gene allele is usually the one that is expressed. So for example, if I have a skin color that is dominant, such as a dark skin color, if I get married to somebody that is white, our child is going to have the dominant skin color, which is going to be black. And that is what is going to be what is expressed. So the phenotype is going to be the skin color that is observed, but then the allele that is expressing the skin tone or skin color is going to be the genotype. And because that is the dominant genotype, being the dark skin, that is what is going to be expressed, and that is why my child is going to be black. Chromosomal abnormalities are a major cause of fetal loss, and that is why we are talking about genetics, because it is important in reproduction, and it also causes congenital problems and gynecologic disorders. Abnormalities can occur during mitosis, which is the division of somatic cells, or meiosis, which is the division of the sex cells. These abnormalities are due to deviations from the correct number of chromosomes per cell. As I said, the human cell has 46, 23 pairs, and so if there are any numbers that are different, whether it's lacking or more than the 46, then there are going to be some genetic problems. So when there are more or less or missing parts of the chromosomes, then a genetic condition will result from that. So this is an example of the genes and the pairs of the genes. These are some examples of disease processes that result from the different types of chromosomal abnormalities. And we are not going to go into a lot of details about the symptoms of this disease. Just know that these are some examples. So for example, if there are any abnormalities with the number of chromosomes, then you can have Down syndrome. Abnormalities with the structure of the chromosome can result from translocation, where the gene is attached to a section on the genome that is not supposed to, or where there is a removal of that particular gene from a spot that is supposed to occupy. And like I said, we won't go into much details. So this is an example of how the gene is going to be expressed, and this is what we call the genetic key when you are constructing a genogram. Sex chromosome abnormalities are going to result in what we call Turner syndrome, and that is monosomy. That means that only one of the genes was acquired by the infant or the fetus. And then you have the Klinefelter syndrome, which has more than needed, and so they have a double X gene, and so this causes some disease because they have more than they need. There are also unifactorial, which is a single gene controlling a trait or disorder. So genes can be inherited in one of the following ways: either autosomal dominant, which is a single gene trait that requires the gene alleles controlling the trait to be located on an autosomal chromosome. The trait appears in every generation with no skipping. So with autosomal gene expressions, if they are dominant, you are going to see the gene expression in all generations with no skipping. So it means that your grandfather had it, your father had it, and you are going to also have it. The risk for an affected adult to pass the trait to a child is 50% with each pregnancy for this type of gene acquisition. Unaffected adults do not have affected children, therefore their risk is essentially zero. So there is a 50% chance that I'm going to have it, but if I don't get it, then it means I'm not going to transfer it to my offspring, and so my children are not going to have it. However, the trait is found about equally in males and females in the family, and an example of this would be diabetes mellitus 2. So diabetes type 2 can affect the males and females in the family regardless of whether they are adult or whether they are young, and this is because it's an autosomal dominant inheritance, and this does not skip generations. So it means that if my mother has it, then I have a 50% chance of getting it. Okay. Penetrance is how often or how well within a population a gene is expressed when it is present. So a highly penetrant gene has high probability of being expressed, and expressivity is the degree of expression a person has when a dominant gene is present. And so in autosomal recessive in autosomal dominant inheritance, the gene is very penetrative and its expressivity is high. That's why it's at 50%. Okay. Then we move on to autosomal recessive. The trait may not appear in all generations because it's recessive. Remember, we said the dominant trait is what is expressed. So for this to express, you actually need two genes from both parents for it to be evident or for it to be expressed. It appears in all generations of any one branch of a family. So the trait often first appears only in siblings rather than in parents and children, and about 25% of the family will be affected and express the trait. I don't want you to go into very much details about this because we just want you to know something about these. Okay. Then we have the sex-linked recessive inheritance, and the X-linked recessive genes have dominant expression in males and recessive expression in females. This is because when you have a baby boy or a fetus that is a male, they inherit an X chromosome from the mother and a Y chromosome from the father. And so if you have a gene that is expressed on the X chromosome, then they don't have another gene to kind of counteract, and so they are going to be automatically expressed in the male because they don't have the other gene to kind of balance it. So this difference in expression is because males do not have a second X chromosome to balance the presence of a recessive gene on the first X chromosome. Sex-linked or X-linked recessive single gene traits require that the gene allele be present on both of the X chromosomes for the chromosome for that particular trait to be expressed in females. So if I have a female daughter, it means that I should have the gene, my wife should have the gene for them to be able to express if the gene is a recessive gene. The incidence of the trait is much higher among males in a family than among females, as I explained earlier on, because the gene is not does not have any other allele to counterbalance when you have a male who has an XY chromosome. So transmission of the trait is from father to all daughters who will be carriers, and then last female carriers have a 50% risk with each pregnancy of passing the trait to their children. Okay, and an example of this is your hemophilia. There is also the multifactorial aspect of genetic transmission, and as we said, genetic disease transmission can be affected by many other factors in the environment, such as radiation pollution, such as exposure to certain chemicals. Many gene defects can be attributed to multiple factors stemming from individual genetic makeup and the effect of the environment. Examples of disease that have multifactorial variations include cleft lip palate, which means it's caused by multiple factors. We cannot really pinpoint one particular thing that causes that gene mutation. And also congenital heart disease, neural tube defects, pyloric stenosis. We will talk about some of these when we discuss care of the newborn and children. So now, when we were talking, we said as a nurse, you should be able to develop a pedigree or a genetic map for your client. So what is a pedigree? Is any pattern of inheritance recognized, or the trait may be expressed in certain individuals in the family, and you are just putting this map together to be able to determine, is any pattern of inheritance recognized, or does the trait appear sporadic? Is the trait expressed equally or unequally among males and females in the family members? And is the trait present in every generation, or is this skipping one generation and then appearing in the next generation? Or do only affected adults have children who are affected with the trait? And once we know this, then we will be able to determine the pattern of inheritance and to be able to cancel or determine the risk for our client. But before we can draw a pedigree or do a genogram, we have to know the symbols on the genogram. So with every pedigree that you see, there are going to be keys that are developed, and this key is going to be similar in certain instances, but they can also vary depending on your facility. So you just have to use the key to be able to determine what is in the pedigree. And for the purpose of this class, this is the key that came with your book, and this is what we are going to use. Okay. So let's talk now about genetic counseling. Genetic assessment and counseling is a routine in medical practice. As I said, it's recommended that you obtain genetic information, such as the family history, with every admission, and this is because we want to be able to counsel our patients. The National Institute of Health recommends obtaining this genetic or family history, and this is not a mandate, but it's highly recommended, and most hospitals do get genetic history when you visit them. In obstetrics, the goal is to identify risk and provide interventions such as counseling, or to facilitate the planning of the care for this child, to prepare the parents for them to be aware of the genetic defects and to take necessary steps to be able to mitigate the effects of the genetic disorder. Genetic testing has many benefits due to its ability to confirm diagnosis or identify at-risk individuals. However, the nurse should be aware that genetic testing results do not change and hence genetic disease cannot be cured, at least not for now. There are many tests that are being developed to help with proper diagnosis and treatment of certain genetic conditions, and there are genes that are being mutated to see their effect, but in many cases the advancements have not reached a point where we can cure some of the genetic conditions or any of the genetic conditions as at now. And so we should be aware that once the results are proclaimed by the provider, it cannot be changed, and so the individual will have to live with that. So we should be thinking about the psychosocial, social risks such as the stress, anxiety, depression, and the risk for family disruption that comes with genetic testing. The goal of counseling is to communicate the risk occurrence before conception or reoccurrence after birth. So if a client has had a baby or a child that has genetic conditions, then we should be counseling them to prevent the recurrence, to let them know, okay, if you have another baby, you still have this chance, maybe 50%, 75%, or 25% chance of having another baby. We should note that in most cases, when we say 50%, it doesn't mean that if you have 4 children, 2 of them are going to have that genetic condition and 2 of them are not. What it means is with every conception, with every child, you stand a 50% chance of the child having a genetic condition. So we should make that pretty clear to the parents. The provider should also provide a clear interpretation of present and future risk related to the genetic disease. And remember that genetic counseling is a process. Advocate for your patient and provide patient-centered care by establishing rapport with the patient and listening to the patient concerns and working to address that. Be part of the collaborative effort to help the patient. So as you work with the interprofessional team, your role as the nurse is to be able to alleviate the patient anxiety, to give them as much information as you can and to direct them to resources. So be non-directive when you are giving genetic counseling. The decision comes from the patient. So if I know I have a risk for sickle cell, or if I am a carrier and therefore my children are at risk for developing sickle cell, it is my decision whether I want to have children or not. You as the nurse cannot tell me not to have children. So be non-directive, but avoid also giving your opinion, and ensure that clear communication and information that is given is accurate, so the client can make an informed decision. Then observe the ethics and provide support in the decision-making process. So if I am going to go for genetic testing, what are the ethical situations around that genetic testing? You as the nurse should be well versed in it to let me know, so I will be able to make that decision with all the information that I have. And then privacy is a major concern. Maintain privacy and confidentiality, because genetic information, just as any medical record, is protected by HIPAA. And so we have to be aware of all these as we give genetic information. So the question is, a patient is waiting or awaiting the results of genetic testing. Which patient statement requires immediate nursing intervention? If you said C, then you are right. The nurse should always be aware of patient indication that he or she has a degree of psychological, psychosocial stress or instability, or could accomplish self-harm. So as we said, Genetic testing and results can put a lot of stress on the patient. If a patient expresses that they don't know what they will do if the results are positive, that is an indication of a psychological or psychosocial stress, and that can lead to them harming themselves. And so it is important that the patient understand and be prepared for possible psychological, social, and or familial risk. In this scenario, the nurse should immediately intervene and investigate by following up with other questions such as, do you intend, or what do you intend to do? How are you going to react? What are your plans if this is positive versus if it's negative? Not knowing what the results will be can incite self-anger, and that can lead to self-harm. So you want to be sure that you mitigate this as a nurse. The next question asks you, which patient would be considered to have the greatest genetic risk for developing a disease or disorder? And so you are looking at multiple factors. So if you chose B, then you are right, because breast cancer at an early age can indicate a genetic predisposition. So a 38-year-old female whose mother died of breast cancer at 40 means that this particular client is at a higher risk for breast cancer herself. So this patient's mother had the cancer at an early age, and that predisposes the 38-year-old who has just 2 more years before she turns 40 to cancer development. So thank you for listening to this recorded session. Make sure you do the readings and that you are ready for the in-class activities to help you understand this concept. This was just a brief presentation. Genetics is a very large field, but we just have to have a basic understanding of what goes on in genetics as we provide care. So the main goal for us knowing about genetics is to be able to draw the family pedigree and to determine the pattern of inheritance. And we are going to do some activities in class that will give you the opportunity to practice the mapping or the drawing of a pedigree or genogram. Thank you and see you in class.